Human embryology is the remarkable story of our very beginning. It is, honestly, the science of how a single fertilized cell transforms into a complex, multicellular human being over a span of just 8 weeks. This field is not merely an academic exercise, it is the fundamental blueprint of human life. It reveals the intricate processes that build our bodies, from the first division of cells to the formation of every single organ. Understanding this journey provides a profound insight into human anatomy, physiology, and the origins of many congenital conditions. For any aspiring doctor, this knowledge is the bedrock upon which a deep understanding of medicine is built. This intricate dance of development begins with a single event, but unfolds through a cascade of precisely timed molecular and cellular interactions. Think of it as a highly sophisticated construction project, where cells are the workers and DNA is the master plan. Each cell must know where to go, what to become, and when to act. This process follows a universal timeline, yet it is unique for every individual. As we delve into the stages of embryology, we uncover the secrets of how our complex structures, like the brain, heart, and limbs, are meticulously sculpted from the simplest of beginnings. It is a story of incredible biological engineering. Studying embryology allows us to appreciate the sheer complexity and elegance of human development. For instance, did you know that during the 3rd week of development, the embryo is no larger than a poppy seed, yet it is already laying down the foundations for the entire nervous system? These early weeks are a period of breathtakingly rapid change, where the basic body plan is established. Errors in this delicate process can lead to significant structural anomalies, which is why understanding the normal sequence of events is so crucial for diagnosing and managing health conditions that originate before birth. Ultimately, embryology connects the microscopic world of genes and cells to the macroscopic world of anatomy that you will study in the dissection lab. It answers the fundamental questions of how we are made. Why is the heart on the left side? How do our fingers separate from a paddle-like handplate? Why do certain nerves follow such convoluted paths? The answers lie in the developmental journey our bodies undertake. For future clinicians, this knowledge is not just fascinating, it is indispensable for understanding human health and disease from the very first moment of life. The story of a new human life begins with fertilization. This is the moment a sperm cell successfully penetrates and fuses with an oocyte, or egg cell. This remarkable event typically occurs in the fallopian tube. Out of millions of sperm that begin the journey, only one will succeed. Upon fusion, the sperm releases its genetic material into the oocyte. This combination of genetic information from both parents creates a single, unique cell called a zygote. This new cell contains the complete set of 46 chromosomes, which is the entire genetic blueprint required to build a new individual. This moment marks the true initiation of biological development. Once the zygote is formed, it embarks on a journey of rapid cell division known as cleavage. While traveling down the fallopian tube towards the uterus, the single cell divides into 2, then 4, then 8, and so on. This happens without the overall size of the embryo increasing, so the cells, called blastomeres, become progressively smaller. By about the 3rd day, this cluster of cells forms a compact ball called a morula, resembling a tiny mulberry. It is a solid sphere of cells, each one with the potential to contribute to the developing human. The journey to the uterus takes several days, and all the while, this process of division continues relentlessly. Around the 5th day after fertilization, a crucial transformation occurs. The morula develops a fluid-filled cavity, turning it into a hollow structure called a blastocyst. This is a critical stage, as the cells begin to differentiate into 2 distinct groups. The outer layer, known as the trophoblast, will later develop into the placenta, the vital organ that nourishes and protects the growing embryo. The cells on the inside form a cluster called the inner cell mass, or embryoblast. It is this inner cell mass that holds the incredible potential to form all the tissues and organs of the future baby. The final step in this initial phase is implantation. Arriving in the uterus around day 6 or 7, the blastocyst must attach itself to the uterine wall, the endometrium. The trophoblast cells play an active role here, burrowing into the uterine lining to establish a secure connection. This process is essential for the embryo to gain access to the maternal blood supply for oxygen and nutrients. Successful implantation marks the end of the 1st week of development and the beginning of a more complex phase of growth, establishing the pregnancy and setting the stage for the dramatic changes to come. Following implantation, the embryo undergoes one of its most critical and dramatic transformations, gastrulation. This process, occurring during the third week of development, is where the two-layered disc of the inner cell mass reorganizes itself into a three-layered structure. It is often said that gastrulation is the most important time in your life. It is the moment the fundamental body plan is established. The process begins with the formation of the primitive streak, a groove that appears on the surface of the embryo. Cells from the top layer migrate inwards through this streak, creating three distinct germ layers. These three primary germ layers are the ectoderm, the mesoderm, and the endoderm. Each layer is fated to give rise to specific tissues and organs as development progresses. The ectoderm is the outermost layer, and it is responsible for forming the structures that interact with the outside world. This includes the entire nervous system, both the central, meaning the brain and spinal cord, and peripheral components. It also forms the epidermis, which is the outer layer of the skin, as well as our hair and nails. Essentially, the ectoderm builds our external surface and the system that allows us to sense and respond to our environment. The middle layer, the mesoderm, is arguably the most diverse. It is responsible for generating the majority of the body's structural components. This includes all types of muscle, skeletal, smooth, and cardiac. The skeleton itself, meaning bone and cartilage, and the circulatory system, including the heart, blood vessels, and blood cells. The mesoderm also forms the dermis, which is the inner layer of the skin, and the excretory and reproductive systems. It is the layer that provides the body with its framework, its ability to move, and the means to transport vital substances throughout the body. Finally, the innermost layer is the endoderm. This layer primarily forms the lining of the body's internal tubes and passages. It gives rise to the epithelial lining of the entire digestive tract, from the pharynx to the rectum. It also forms the lining of the respiratory tract, including the trachea, bronchi, and lungs. Furthermore, vital glands that originate from the digestive tube, such as the liver, the pancreas, and the thyroid gland, are all derivatives of the endoderm. In essence, the endoderm creates the machinery for processing food, absorbing nutrients, and exchanging gases with the environment. With the three germ layers established, the embryo enters the period of organogenesis, which spans from the end of the third week to the end of the 8th week. During this time, the flat layers of cells begin to fold, migrate, and differentiate to form the rudimentary organs. One of the very first systems to develop is the nervous system. The ectoderm thickens to form the neural plate, which then folds to create the neural tube. This structure will become the brain and spinal cord. This process, called neurulation, is a prime example of how a simple sheet of cells can give rise to a highly complex and vital organ system. Simultaneously, the mesoderm and endoderm are also hard at work. The heart begins as a simple tube that starts to beat and pump blood around the 22nd day after fertilization, often before the mother is even aware she is pregnant. The mesoderm on either side of the neural tube organizes into blocks called somites, which will form the vertebrae, ribs, and skeletal muscles. Meanwhile, the endoderm folds to form the primitive gut tube, the precursor to the digestive tract. By the end of the 8th week, all major organs and body systems have been established in a recognizable, albeit immature, form. This period is incredibly sensitive to disruption. Because so many fundamental processes are occurring simultaneously, the embryo is highly vulnerable to genetic abnormalities and external factors like certain medications, infections, or toxins. These are known as teratogens. Exposure to a teratogen during this critical window of organogenesis can lead to major congenital malformations. For example, defects in neural tube closure can result in conditions like spina bifida. This highlights why understanding the precise timing of development is so clinically relevant. It helps explain the origins of birth defects and informs prenatal care. For you, as a future medical professional, this knowledge is not just academic. It is the foundation for understanding pediatrics, obstetrics, and even adult medicine. When you see a patient with a congenital heart defect, your knowledge of embryology will help you understand why the defect occurred. When you learn about the anatomy of the nervous system, you will appreciate its developmental origins. Embryology provides the why behind the what of human anatomy. It transforms the study of the human body from a task of memorization into a logical story of development. making you a more insightful and knowledgeable clinician.